Definitions of cancer. Cancer defines as uncontrolled growth of abnormal cells and the ability of these cells to migrate from the original sites and spread to the distant site. How cytogenetic tests can detect cancer cells? Cytogenetic tests can detect genetic alterations, mutations in the DNA that control cell growth and proliferation. Nowadays, cytogenetic tests has been used to diagnose breast cancer, acute lymphoblastic leukemia, acute myeloid leukemia, renal tumor, and many more. Two types of tests in cytogenetic for cancer diagnosis. First, conventional method, and second, molecular method. In conventional method, we can do karyotyping analysis. Laboratory procedure that allow examinations of a patient's set of chromosomes. Karyotypes allow refer the actual collections of a chromosomes being examined. Examining these chromosomes through karyotyping allow your physicians whether there are any abnormalities or structural problems. Breast cancer cell line karyotypes analysis shows a polyploidy, as shown in the picture. In molecular method, we can do fluorescence in situ hybridization FISH. It's procedure that allow researchers to locate the positions of specific DNA sequence on the chromosomes by using fluorescent probes as a diagnosis for many types of chromosome abnormalities in the patient. This picture shows a normal interface cell of genes ERBB2 in the green color and the CEN17 in orange color. And this picture shows a breast carcinoma tissue sections, shows the ERBB2 genes cluster in a green color and CEN17 in orange color.
Cytogenetics from 1,000 feet up, flow cytometry from 500 feet up, FISH, which dropped us right onto the street to be able to look at all of the doors and windows or genes, and molecular technology that allows us to look at DNA base pair sequences. Prior to this technology, FISH, fluorescence in situ hybridization, developing, we had no way to look or touch a gene. It was a concept, it was a theoretical. We understood it existed, but it was a cause and effect. FISH gave us the ability to light these genes up. FISH gave us that ability to look at and to touch genes and answered the next generation of questions for probably 20 years.
Second method, comparative genomic hybridization CGH and array comparative genomic hybridizations ACGH. It modified in situ hybridization technique which allow detections and mapping of DNA copy number variations CNV between two genomes. This technique that compare and measure difference in the copy number variations between two DNA samples, the test and control samples, and allow provide a map of chromosome regions that again or lost. This picture shows a summary of genetic imbalance detected by comparative genome hybridizations in 34 invasive ductal breast carcinoma tumor samples. Vertical line on the left side of each chromosome diagrams represents a loss of genetic material in the tumor, whereas those on the right corresponds to a gain. The number above the bar refer to the case numbers. Each of the cytogenetic tests have their own advantage and disadvantage. In karyotypes, it provides a global information in a single assay, including primary and secondary anomalies, and provide a directions for a molecular study of a petrogenetically important genes. FISH can be applied to both dividing and non-dividing cell. The technology is straightforward. Hybridization with the multiple probe enable detections of translocation product and can identify a range of mutations. CGH is cheaper than ACGH, a global scan of the whole genome without the need for the specific probes and high throughputs, high resolutions than fish and karyotypes. ACGH shows a high resolution detections of small changes, easy, faster, less technically demanding. Disadvantage of cytogenetic tests. In karyotypes, it requires a fresh tissue. Some microscopic or cryptic rearrangements may result in a false negative result and require a long time to prepare. In fish, it cannot detect a small mutation, misuniparental disomy, misinversions. Probes are not yet commercially available for all chromosomal regions. In CGH, balance translocations undetectable, low resolutions cannot detect microdeletions and microduplications, and ACGH balance translocations are undetected and low sensitivity. Thank you.